Hello, my name is Jamie Simpson, and today we're going to be talking about the importance of the polyadenylation signal in RNA cleavage and the relevant data that supports this. This video was made for MCDB 427 Molecular Biology at the University of Michigan. Enjoy! First off, let's start with a general overview of what exactly a polyadenylation signal is. We know that eukaryotic RNA is different from prokaryotic RNA since, in eukaryotes, several modifications will happen to the RNA either co-transcriptionally or post-transcriptionally. A few of these modifications include intron splicing, addition of a 5' cap to the RNA, and the addition of a string of A nucleotides called the poly-A tail to the 3' end of the transcript. These modifications most commonly take place on mRNA, but other RNAs made by the eukaryotic cells can also be modified. For the purposes of this video, however, we are going to be looking specifically at the poly-A sequence, which is not present in tRNAs or rRNAs. The poly-A tail is present at the 3' end of a mature RNA transcript and is added post-transcriptionally by a specific kind of RNA polymerase known as poly-A polymerase, or PAP. Though there is no specific string of T's coding for the poly-A tail, there is a small 6-nucleotide sequence called the poly-A signal within the transcript that signals for this modification. This sequence has a few different forms, but the general consensus sequence goes by AAUAAA in mammals and plants. Other hexamers of this sequence are used, but are generally not as efficient as the consensus sequence. Downstream of the poly-A signal, there are also two other motifs that are important: the GU-rich stretch and the U-rich stretch, which assist in the formation of the poly-A tail, but we won't be discussing those in this video. In previous research, it was discovered that transcription of eukaryotic genes extended well beyond the polyadenylation site and then was cleaved around 20 to 30 nucleotides downstream of this signal. The RNA that was to become the mature transcript was then polyadenylated at the 3' end created by the cleavage, and the extra RNA that was not needed, it was just degraded. They did not determine how far past the poly-A signal transcription continued, just that it did happen past this signal. In 1981, researchers Molly Fitzgerald and Thomas Shank wanted to look at the importance of this conserved poly-A signal in determining the proper cleavage site of the RNA and the subsequent polyadenylation of the transcript. To test this, they produced recombinant SV40 viral DNA, which had two poly-A signals, and wanted to see how long the resulting transcript was by either including both of the two signals or by knocking out one or the other of the two signals. To measure the length and relative abundance of the fragments, they performed an S1 mapping assay, which, if you aren't too familiar with or just want to brush up on, I'll talk about now before we really get started with the data. S1 mapping is a type of restriction mapping that allows us to determine either the 5' or 3' end of an RNA transcript relative to the position of a known restriction site by hybridizing it to a labeled DNA probe complementary to the RNA of interest. In this experiment, since we're interested in looking at the 3' end of the RNA, the researchers are specifically going to be using 3' S1 mapping, which I'll go over right now. In S1 mapping, we start out with double-stranded DNA, in this case our recombinant SV40 viral DNA, containing several restriction sites for different restriction enzymes to cut. Researchers are then going to cut the DNA, which is going to result in a fragment that contains part of the gene of interest and 5' overhangs. The next step is going to be to fill in those overhangs with a DNA polymerase reaction using all 4 DNTPs, but only one of them is going to be labeled. For example, alpha-32-phosphate labeled DATP. It is important that the labeled phosphorus is on the alpha phosphate since we want it to be incorporated into the DNA and not given off as a pyrophosphate. We also want to label the 3' end of the DNA since we're trying to find the 3' end of the RNA that will hybridize to it. S1 also works to map the 5' end as well. You just need a 5' end label, and there's a slightly different mechanism. After labeling the 3' end, the fragment is then cut with a second restriction enzyme, resulting in 2 fragments of different lengths, which are then run on electrophoresis gel to be purified and to obtain the DNA fragment that will eventually become our probe. After purification, the DNA probe is then denatured from its unlabeled complementary strand and then hybridized to the RNA of interest. Next, a specific kind of nuclease called S1 nuclease is going to be introduced. S1 nuclease will cleave free ends of the single-stranded RNA or single-stranded DNA that is not hybridized, leaving only a double-stranded DNA-RNA hybrid. Since the RNA is going to have a 5' overhang, and the DNA probe is going to have a 3' overhang, the S1 nuclease will chew the free probe DNA and the free RNA until it reaches the end of the RNA-DNA hybrid. The hybridized RNA DNA is then run on electrophoresis gel with molecular size standards so that we can accurately determine its size. The size given indicates that the 3' end of the transcript is located however many nucleotides downstream from the restriction site for the first restriction enzyme that we used. Remember, in this example, the first restriction enzyme that we used was HIN-D3. The 3' end of the RNA is located 475 nucleotides downstream from the HIN-D3 cleavage site, which is located at the left-handed end of the probe. So, in this experiment, the researchers used 3 different types of recombinant SV40 DNA in which they added a second poly-A signal and then cloned. As we can see, for all 3 of the DNA templates, they have all been cleaved by the restriction enzyme PST1 to make sure that any variation in length is at the 3' end. 1471 template DNA contains the 2 poly-A signals intact. This will act as a control to see if one poly-A site is used more preferentially than the other. The other 2 template DNAs, called 1474 and 1475, we can see that they knocked out one or the other of the 2 poly-A signals by making a 16 nucleotide deletion. Also note that there are 2 fragments that we would expect to see if both of these sites were used equally, one of length 680 nucleotides and the other of length 920 nucleotides, leaving around 240 nucleotides difference between the 2 fragments. So, when they performed S1 with the DNA to find out how long the mRNAs were, what did they find? Well, the results looked like this. So, what can we see from this gel? Well, first let me orient you a bit. See those numbers across the top? The first number before the slash is the type of probe that they used, either for wild-type DNA, 1471, 1474, or 1475. The second number is the type of mRNA that they assayed, which is either mRNA created from the DNA template or this thing called UNINF. This stands for uninfected, since these cells were not infected with the SV40 DNA and thus do not contain the recombinant DNA, so we shouldn't expect to see the probe hybridized to anything here. The far right lane is a molecular weight standard, which is used to compare the sizes of known fragments to our fragments to see what size our fragments are. So let's interpret the data a little bit. In the first lane, we see that this is just a wild-type DNA probe assaying for wild-type mRNA. There are 3 bands in this lane that I want to focus on, and one that we will come back to a little later. The bands I want to focus on are these 2 right here, and this one right here. As we can see, there is a band that is around 680 nucleotides. This band corresponds to the mRNA that was cleaved at the original cleavage site, which is located here. Remember that they added this second poly-A signal to the wild-type DNA to create 1471, which is shown here. Because the wild-type DNA does not have the second poly-A signal at 920 nucleotides, this is what we would expect to see, a band corresponding to a fragment of length 680 nucleotides. We also see 2 other bands that ran a little bit smaller than the 680 nucleotide fragment. These are just artifacts that we can ignore. So overall, in lane 1, we get what we would expect. Lanes 2, 3, and 4 all contain the recombinant DNA and are probed with the appropriate probe for the mRNA expressed from that template DNA. Again, ignore these bands at the top. We'll come back to them later. As we can see, in lane 2, the DNA had 2 poly-A signals, and we can see that the 920 nucleotide and 680 nucleotide fragments are present in relatively equal amounts. In 1474, when we knocked out the first poly-A signal at 680 nucleotides, we see that we get a band corresponding to a fragment that's 920 nucleotides long. In 1475, when we knocked out the second poly-A signal, we see that we get a band corresponding to a fragment that's 680 nucleotides long. It's weak, but it's there. So, going back to these bands at the top, as we can see, they run rather large and are relatively strong in intensity. So, what are these? Well, to answer that question, we have to go back to the method of S1 mapping. In S1 mapping, remember that we labeled one end of one of the strands of the double-stranded DNA, but then we had to denature the double-stranded DNA so that we could hybridize the labeled DNA probe to the RNA. Well, since there's no way to separate the labeled DNA strand from the unlabeled DNA strand, because they both have the same size and are relatively equal in charge, this band is due to the labeled probe rehybridizing to the unlabeled complementary DNA. If you're wondering why the wild-type band in lane 1 runs like it's smaller than the experimental bands in lanes 2 through 4, it's because remember that the wild-type DNA does not contain the duplicated poly-A signal, so the DNA itself is shorter than the experimental probe for 1471, 1474, and 1475. So going back to the gel, let's take a look at lanes 5 through 8. We see that these lanes are uninfected cells, so they do not contain the SV40 recombinant DNA, and thus will make no RNA that will hybridize with the probes. This is another set of control lanes with which we can compare to lanes 2 through 4. Looking at these lanes, we see that there are only strong bands at the top, which is where we saw the rehybridized DNA probe and the complementary DNA. In lane 5, there's a little fragment that's around 447 nucleotides in length, but that is the same length as the artifact in lane 1, so we can ignore that. There's nothing in the genome that hybridizes to the S1 probe. Now that we have the data, what does it all mean? Well, there are a few conclusions that we can make from this data. Looking at lane 2, we see that cleavage does not always occur at the first poly-A signal. This is reasonable to assume, since we're talking about protein machinery attaching to the RNA and cleaving it, so it would make sense that these sites are utilized equally by the free-floating protein that will randomly bind to either site. When we knock out either of the 2 poly-A signals in lanes 3 and 4, we get the size fragment corresponding to what we would expect. So what have we learned from this data? Why is it important? Well, we know that the poly-A signal is important for mRNA cleavage, since we saw that transcription will occur past this site and will not be cleaved until the signal is reached. We can also say that the first polyadenylation signal that is present might not always be where the cleavage takes place, since we saw that with 2 poly-A signals, cleavage happened around equally at each site. We can't tell specifically that the poly-A signal is absolutely required for cleavage, since we would have to completely knock out both signals and determine the length of the mRNA. You can't say it's necessary because it's possible that in 1474 and 1475, the intact sequence is outcompeting the knocked out sequence, even though it's not entirely necessary. Thank you so much for watching, and I hope this video cleared up some confusion on this subject.